Assalamualaikum friends, welcome to medzukhruf. Today we are going to talk about Streptococcus. We've got a lot to cover in today's lecture, but before starting the lecture, I'd like to tell you guys that these videos are meant for educational purposes. Things and treatment may change with time. If I get wrong or miss anything, your input is always welcomed in the comment section. So let's dive straight into the video. Streptococcus. It is a gram-positive bacterium. I've got a detailed video on gram staining. If you want to watch it, find this link in the description. It has got two words in it. First one is strep, which means chains, and the second one is coccus. Coccus means spherical. Spherical is kind of round. So this bacterium is the one that forms chains. These chains can contain pairs, and this bacterium resembles a string of beads. As you can see in this picture, this bacteria exists in chains, and these chains contain pairs as well. In some places you might not find pairs, but in this picture you can see this bacterium in pairs. This bacterium belongs to the family Streptococcaceae and is catalase negative. If you remember from my Staphylococcus lecture, Staphylococcus is a catalase positive bacterium, but this one is not. Why? We're going to talk about that in the lab diagnosis section. Classification of this bacterium is based on different variables, but here we're going to look at the oxygen requirement. It is classified based on oxygen requirement into facultative, anaerobe, aerobes, and anaerobe. This bacterium is non-motile because it has no motility apparatus, as you can see in this picture, like a flagellum. Lecture outline, we are done with the introduction of Streptococcus. Now we'll be looking at its classification, which is really important. Then we'll talk about its morphology, habitat, transmission, pathogenesis, clinical findings, lab diagnosis, treatment, prevention, and at the end, as usual, we'll review the lecture. Classification. Streptococcus is further classified on the basis of serology. That's also called the Lancefield classification. Sero is the word derived from serum, and logy is for studies, so it is the study of serum. It is also classified on the basis of hemolysis and biochemistry. And in hemolysis, heme is the word that refers to blood, and lysis is the breakdown. So the breakdown of blood is hemolysis. Biochemistry refers to the reactions occurring in living organisms, and we study them. On the basis of serology, Streptococci are arranged into groups from A to W, but in some places you also find U. So for UEs, I've mentioned W and U both there. This classification is devised by Rebecca Lancefield and is also called as Lancefield classification, and these groups are also termed as Lancefield groups. Bacteria are arranged in these groups on the basis of antigenic differences in C carbohydrates. Don't worry, we're going to talk about that carbohydrate in morphology section. Group A Streptococci, for example Streptococcus pyogenes, are one of the most important human pathogen. The group B Streptococci, for example Streptococcus agalactia, they're also very important. They colonize the genital tract of females, and they are responsible for causing neonatal meningitis and sepsis. We're going to talk about all of that stuff in today's video. Then we've got group D, that is important. It includes Enterococci, for example Enterococcus faecalis and Enterococcus faecium, and also non-Enterococcus bacteria, for example Streptococcus bovis. The group C, E, F, G, H, and also from K to U are the Streptococci that are infrequently responsible for causing human diseases. On the basis of hemolysis, the Streptococcus is classified into alpha, beta, and gamma. Alpha hemolytic bacteria, beta hemolytic, and gamma hemolytic bacteria. The alpha hemolytic bacteria are further classified on the basis of apoptogen disc present in a petri dish. If a bacteria is sensitive to it, it is the Streptococcus pneumoniae, which is a capsulated bacteria, and if a bacteria is resistant to that apoptogen, it is the Streptococcus viridans. It is non-capsulated bacteria. In some places, this word apoptogen is also pronounced as apoptogen, so you should choose the one you like. How we'll find that the bacteria is sensitive to apoptogen or resistant to it? As in this picture, you can see that these are the petri dishes, and in them, these are the bacterial colonies, and in the center, you can see the apoptogen disc. A bacteria being sensitive to apoptogen will form empty area around the apoptogen disc, which means that this bacteria is killed with that disc. Apoptogen is a quinine derivative, so Strep pneumoniae is sensitive to that. And if we look at the right side, there are no empty spaces around that disc, which means that this bacteria survived in the presence of apoptogen disc. This is Strep viridans. The beta hemolytic Strep are further classified based on antigenic structure. This is also the Lancefield classification. Serology test based on the carbohydrate antigen of the cell wall of bacteria, as we've discussed. If a bacteria is sensitive to bacitracin is an antibiotic, so if a bacteria is sensitive to it, it is group A bacteria, for example Streptococcus pyogenes, and if a bacteria is resistant to it, it is group B bacteria, for example Streptococcus agalactiae. As in this picture, you can see that group A bacteria, for example Streptococcus pyogenes, is sensitive to bacitracin disc because it is killed in its presence, and we can see the empty area around that disc. And the group B, for example Streptococcus agalactiae bacterium, is resistant because it survived in the presence of bacitracin disc, and there are no empty spaces we can see around that disc. And the gamma hemolytic bacteria is further classified on the basis of 6.5% salt solution. If a bacterium grows in that solution, it is Enterococci, which is further classified into Enterococcus faecalis and Enterococcus faecium, which belongs to the group D Streptococcus in the Lancefield classification. If there's no growth observed in salt solution, that is non-Enterococci, for example Streptococcus bovis, it was formerly called Streptococcus bovis, but now it is called as Streptococcus galolyticus. As we've talked about, that on the basis of hemolysis, bacteria are further classified into alpha, beta, and gamma. We can also classify for or ease in order to understand the classification of strep bacteria. On the basis of hemolysis, it's classified into beta hemolytic and non-beta hemolytic. The beta hemolytic are also called the Lancefield bacteria, or the Lancefield classification, which includes the serologic classification from group A to U or W. For example, group A, beta hemolytic streptococci. And in non-beta hemolytic bacteria, we've got alpha and gamma. For example, the Viridans streptococci is not a bacterium, it is a group of different bacteria. For example, Streptococcus mitis, Streptococcus sanguinis, Streptococcus mutans, Streptococcus intermedius, and Streptococcus aeruginosus. There's one more strep bacteria that is really important, and it is called Peptostreptococci. It's further classified into Peptostreptococcus magnus and Peptostreptococcus anaerobius. It is the part of normal flora of human being. We are going to talk about them later in today's video. Biochemical test depends on the physiological properties of the Streptococci. Morphology. Shape. Streptococcus is spherical, round to ovoid in shape. Arrangement is arranged in chains or pairs, but these pairs might be present in chain. They appear like a string of beads, and the chains appear bent or twisted. Its diameter varies from 0.6 to 1 micrometer. It is purple or blue in color. Why? Comment down below. Because it is gram-positive, it will stain purple or blue in the presence of gram stain. As you can see this, strapped under microscope, it is stained purple. It is spherical in nature. It has got no motility apparatus like a flagella. It is existing in chains and pairs. It has got no motility apparatus like a flagella. It has a capsule that can protect it against phagocytosis. It has a cell wall, and the cell wall contains the C carbohydrate and M protein. Both are the beta hemolytic antigens. We are going to talk about them in a moment. This bacteria also has the cell membrane. This bacterium divides along a single axis and is non-spore forming bacteria. C carbohydrate. It determines the group of beta hemolytic streptococci. It is located in the cell wall. Its specificity is determined by an amino sugar. For example, group A beta hemolytic streptococci like Streptococcus pyogenes are distinguished from group B beta hemolytic streptococci like Strep agalactiae because they have a different C carbohydrate. Both the group A and B streptococci have C carbohydrate in their cell walls, but both have different ones. This is the main point to distinguish these bacteria. Next one is M protein. Is the most important virulence factor of strep pyogenes. It protrudes from the outer surface of the cell wall and blocks phagocytosis. That is, it is antiphagocytic. It inactivates C3b, a component of complement that opsonizes the bacteria, prior to phagocytosis. M protein also determines the type of group A beta hemolytic streptococci. There are approximately 100 serotypes based on the M protein, which explains why multiple infections with Streptococcus pyogenes can occur. Antibody to M protein provides type-specific immunity. Strains of Streptococcus pyogenes that do not produce M protein are non-pathogenic. The strains of Streptococcus pyogenes that produce certain M protein types are rheumatogenic, i.e., cause primarily rheumatic fever, whereas strains of Streptococcus pyogenes that produce other M protein types are nephrogenic, i.e. cause primarily acute glomerulonephritis. Although M protein is the main antiphagocytic component of Strep pyogenes, the organism also has a polysaccharide capsule that play an important role in retarding phagocytosis. Habitat. On top of everything, Streptococci are a part of human normal flora. For example, group A Streptococcus is present on skin and in oropharynx. Group B is present in female genital tract and colon. Streptococcus group belonging to group D is present in colon. Streptococcus belonging to viridans group are present in oropharynx or colon. The peptostreptococci are found in gut, mouth, and female genital tract. Transmission. Transmission of Streptococcus occurs when bacteria gain access to tissue or blood by any means, like by surgery or any kind of cut on skin. And it also depends on the type of disease. For example, if it is causing pharyngitis, it can spread to other people via respiratory droplets when a person is coughing or sneezing. Pathogenesis. Group A Streptococci, for example Streptococcus pyogenes, cause disease by following mechanisms. Number 1, pyogenic inflammation, which is induced locally at the site of organism in tissue. Number 2, exotoxin production, which can cause widespread systemic symptoms in areas of body where there are no organism. And number 3, immunologic, which occurs when antibody against a component of organism cross-reacts with normal tissue or forms immune complexes that damage normal tissue. The immunologic reaction causes inflammation, for example, the inflamed joints of rheumatic fever, but there are no organisms in the lesions. Group A strep, for example, strep pyogenes, cause diseases like pharyngitis, skin infection, for example, cellulitis and pyoderma. Let's see how. It causes pharyngitis by adhering to pharyngeal epithelium via pili composed of lipoteichoic acid and M protein. And how is it responsible for causing skin infections? Many of its strains have hyaluronic acid capsule that is antiphagocytic, and antibodies are not formed against this capsule because hyaluronic acid is a normal component of human body. Group A strep also produces certain enzymes, and these are really important for the pathogenesis. Number 1 is hyaluronidase. It degrades hyaluronic acid, which is the ground substance of subcutaneous tissue. Hyaluronidase is known as spreading factor because it facilitates the rapid spread of Streptococcus pyogenes in skin infections like cellulitis. The second one is streptokinase. It is also termed as fibrin and lysine. It activates plasminogen to form plasmin, which dissolves fibrin in clots, thrombi, and emboli. It can be used to lyse thrombi in coronary arteries of heart attack patients. Number 3 is DNA. It is also called as streptodonase, as its name shows that it is going to degrade DNA. DNA is for DNase, and anything that ends with ase is an enzyme. So it is going to degrade DNA in exudates or necrotic tissue. Antibody to DNase B develops during pyoderma. This can be used for diagnostic purposes. Streptokinase and streptodonase, which is DNase, mixtures applied as a skin test give a positive reaction in most adults, indicating normal cell-mediated immunity. The last enzyme is IgG degrading enzyme. IgG is a protein, so this enzyme is a protease that specifically cleaves IgG heavy chains. This prevents opsonization and thereby enhancing the virulence of organism. This bacteria also produces toxins and hemolysin, and these also play an important role in pathogenesis along with those enzymes. These are 5 in number. First one is erythrogenic toxin, second one is streptolysin O, third one is streptolysin S, fourth one is pyrogenic exotoxin A, and the final one is exotoxin B. Let's talk about them. The erythrogenic toxin causes the rash of scarlet fever. Its mechanism of action is similar to that of toxic shock syndrome, toxin type 1 of Staph aureus. If you've missed my videos on Staphylococcus, find their links in the description. This toxin is produced only by certain strains of Streptococcus, lysogenized by a bacteriophage carrying the gene of toxin. The injection of a skin test dose of erythrogenic toxin, the DIC test, gives a positive result in person lacking antitoxin, i.e. susceptible persons. Second one in the list is streptolysin O. It is a hemolysin that is inactivated by oxidation. That's why we've got the letter O after the word streptolysin, which shows that it is oxygen-liable hemolysin, and it causes beta hemolysis only when colonies grow under the surface of a blood agar plate. An antibody ASO develops after group A Streptococcus infections. The titer means the concentration of ASO antibody can be important in diagnosis of rheumatic fever. The third one in the list is streptolysin S. It is also a hemolysin but is oxygen stable. That's why we've got letter S after it. It means that it is not inactivated by oxygen. It is not antigenic but is responsible for beta hemolysis when colonies grow on the surface of blood agar plate. Fourth one in the list is pyrogenic exotoxin A. It is the toxin responsible for most cases of streptococcal toxic shock syndrome. Mode of action of this toxin is similar to that of staphylococcal toxic shock syndrome type 1, TSST. That is, it acts as a super antigen that causes the release of massive amount of cytokines from helper T cells and macrophages. The last toxin in the list is exotoxin B. It is the protease that rapidly destroys tissue and is produced in large amounts by the strains of Strep pyogenes, the so-called flesh-eating Streptococcus. Why? Because it causes necrotizing fasciitis. Till now, we were discussing Group A strep pathogenesis. Now let's talk about Group B strep, like Strep agalactiae pathogenesis. It is based on the ability of the organism to induce an inflammatory response. However, unlike Strep pyogenes, no cytotoxic enzyme or exotoxins are released by this bacteria because there's no evidence for that. And Group B Streptococci have a polysaccharide capsule that is antiphagocytic, and anti-capsular antibody is protective, and it is responsible for hydrolyzing hippurate. Hippurate, also called hippuric acid, is a carboxylic acid. An organic compound is found in urine and is formed from the combination of benzoic acid and glycine. The Group B Strept is responsible for causing neonatal meningitis and sepsis. Pathogenesis by strep pneumoniae and viridans strept is uncertain, as no exotoxin or tissue-destructive enzymes are released by them. The main virulence factor of strep pneumoniae is its antiphagocytic polysaccharide capsule. Many of the strains of viridans streptococci that cause endocarditis produce a glycocalyx that enables the organism to adhere to heart walls. One of its types, the strept mutans, is found in dental plaques and that lead to dental caries. And this infection is spread to brain because of the dental surgeries, as they're found in the normal flora of mouth. So after dental surgeries, these bacteria gain access to brain through the blood vessels and are responsible for causing diseases in brain like brain abscesses. Group D Streptococcus is responsible for causing urinary, biliary, and cardiovascular infections. The Peptostreptococci, what does it do? The Peptostreptococci grow under aerobic or microaerophilic conditions and produce variable hemolysis. And they also participate in mixed aerobic infections. You might be thinking, what are mixed aerobic infections? The term mixed aerobic infections refers to the fact that these infections are caused by multiple bacteria, some of which are anaerobes and others are facultative. For example, Peptostreptococci and viridans streptococci, both members of oral flora, are often found in brain abscesses following dental surgery. Clinical findings. Let's talk about the Group A strep, like Streptococcus pyogenes first. Then we'll talk about Group B, D, and so. Group A causes 3 types of diseases. Number 1, pyogenic diseases, such as pharyngitis is the most common bacterial infection and is commonly called sore throat, And if we call it streptococcal pharyngitis, it is famous with the name strep throat and is characterized by throat pain and fever on examination and inflamed throat and tonsil seen, often with a yellowish exudate, and the cervical lymph nodes are found to be tender. If left untreated, it can lead to inability to swallow and cause other complications like sinusitis, otitis media if it spreads to middle ear. If it spreads to meninges, will cause meningitis. If spreads to mastoids, will cause mastoiditis. And is also responsible for causing skin and soft tissue infections like cellulitis, erysipelas, necrotizing fasciitis. Is also called streptococcal gangrene or eating flesh disease. Impetigo, a form of bioderma, is a superficial skin infection characterized by honey-colored crusted lesions. And lymphadenitis can occur especially on the forearm associated with an infection on hand. The second type of disease is responsible for causing is the toxigenic disease, such as scarlet fever and toxic shock syndrome. If the infecting streptococci produce erythrogenic toxin and host lacks antitoxin, scarlet fever may result. A strawberry tongue is characteristic lesion seen in scarlet fever. Streptococcus pyogenes also causes another toxin-mediated disease, the streptococcal toxic shock syndrome, which has clinical findings similar to those of staphylococcal toxic shock syndrome. However, streptococcal toxic shock syndrome typically has a recognizable site of pyogenic inflammation and blood cultures are often positive, whereas staphylococcal toxic shock syndrome typically has neither a site of pyogenic inflammation nor positive cultures. The third type of disease, the group A strep, is responsible for causing is immunologic disease. For example, rheumatic fever and acute glomerulonephritis. Group B streptococci is responsible for causing neonatal sepsis and meningitis. The main predisposing factor in order for group B strep to cause neonatal sepsis and meningitis is prolonged, longer than 18 hours rupture of membranes in females who are colonized with the organisms during pregnancy. Children born prior to 37 weeks of gestation have a greatly increased risk of disease. Also, children whose mothers lack antibody to group B streptococci and who constantly are born without transplacentally acquired IgG have a high rate of neonatal sepsis caused by this organism. Group B streptococci are an important cause of neonatal pneumonia. Although most group B streptococcal infections are in neonates, this organism also causes infections such as pneumonia, endocarditis, arthritis, cellulitis, osteomyelitis in adults. Postpartum endometritis also occurs. Diabetes is the main predisposing factor for adult group B streptococcal infections. Viridin streptococci, for example, strep mutans, streptomitis, etc. They are responsible for causing infective endocarditis. When they enter bloodstream, they will cause bacteremia. They often enter bloodstream from oropharynx, typically after dental surgery, and are responsible for causing brain abscess. They are also involved in mixed aerobic and anaerobic infections like lung abscesses, abdominal abscesses, for example, liver abscesses. Group D strep includes enterococcal infections like UTIs, endocarditis, intra-abdominal and pelvic infections, and also non-enterococcal infections like endocarditis. Peptostreptococci is responsible for causing abscesses in different parts of body like brain, lung, abdomen, and pelvis. Lab diagnosis. Prior to doing anything else in lab, we need samples. We'll need samples from throat, skin lesions, vagina, and rectum. We'll do this by swabbing. First procedure we'll perform is gram stain. The purple or blue color after gram staining will reveal that this bacteria is gram positive. And microscopy will reveal that this bacteria occurs in chains and chains appear to be bent or twisted. This bacteria is spherical, non-motile, and its diameter is 0.6 to 1 micrometer. As you can see in this picture, its diameter is from 0.6 to 1 micrometer. It is forming chain, it is spherical in nature, and has got no motility apparatus. Catalase test. This test is negative for Streptococcus. Bubbles formation confirm this test. but for this bacteria there will be no bubbles formed. Normally what happens in that test, if a bacteria has a catalase enzyme, it converts hydrogen peroxide in the petri dish into water and oxygen forms bubbles. But as Streptococcus has got no catalase, so it will not convert hydrogen peroxide into water and oxygen, and no bubbles will be formed. So this is a negative test for this bacteria. There are no bubbles formed in this slide and test tube, so this test is negative for Streptococcus. Let's talk about culture. Culture on blood agar will reveal colonies for strep pyogenes, and these will be small, translucent, and beta hemolytic, and these colonies will be revealed in 18 to 48 hours. Diagnosis for streptococcal pharyngitis can be made with the help of a culture. Group B streptococci are characterized by their ability to hydrolyze hippurate and by the production of the protein that causes enhanced hemolysis on blood agar when combined with beta-hemolysin of Staph aureus. The group D streptococci hydrolyze isculin in the presence of bile, i.e. they produce a black pigment on bile isculin agar. As culture tests take a lot of time, there's an alternative available for that test. It is the rapid test. It works by detecting bacterial antigens and it provides results quicker than culture. As you can see there, this is a culture. These are round and they grew in a really uniform way on blood agar. One of the important characteristics for identification of Streptococci is the type of hemolysis. Let's talk about that. The first one is alpha hemolytic bacteria, which means that the bacteria will go for partial hemolysis. The alpha hemolytic streptococci form a green zone around their colonies as a result of incomplete lysis of red blood cells in the agar. The green color is formed when hydrogen peroxide produced by bacteria oxidizes hemoglobin in red color to bilirubin in green color. The second is beta hemolytic. It goes for complete hemolysis. The beta hemolytic streptococci form a clear zone around their colonies because complete lysis of red blood cells occurs. Beta hemolysis is due to the production of enzymes hemolysin called streptolysin O and streptolysin S. We've discussed them. The third one in the list is gamma hemolytic. Some streptococci are non-hemolytic, and these are the gamma hemolytic. If we look at the alpha hemolytic bacteria, it has got green zone around its colony because of partial hemolysis. And if we look at the beta hemolytic bacteria, it has got this clear zone due to complete hemolysis around its colony on the blood agar. And if we look at the gamma hemolytic bacteria, there is no zone, neither green nor clear, because it is responsible for no hemolysis. Serology. ASO is antibody, right? We've talked about them. ASO titers mean their concentrations are high soon after group A streptococcal infections. In patients suspected of having rheumatic fever, an elevated ASO titer is typically used as evidence of previous infection because throat culture results are often negative at the time patient presents with rheumatic fever. Titers of anti-DNase B are high in group A streptococcal skin infections and serve as an indicator of previous streptococcal infection in patients suspected of having acute glomerular nephritis. How do we distinguish strep pneumoniae from strep viridans? They're distinguished by two main criteria. Number one, the growth of pneumonia is inhibited by apoptosin, whereas the growth of viridans streptococci is not inhibited. And number two, that the colonies of strep pneumoniae dissolve when exposed to bile. It means that they are bile soluble, whereas colonies of viridans streptococci do not dissolve. It means that they are not bile soluble, they are bile insoluble. Treatment. Group A streptococcus infections can be treated with either penicillin G or amoxicillin. In mild group A streptococcal infections, oral penicillin V can be used. In penicillin allergic patients, erythromycin or one of its long-acting derivatives, for example azithromycin, can be used. Clindamycin can also be used in penicillin allergic patients. Streptococcal pyogenesis is not resistant to penicillins. Invasive group A streptococcal infections such as necrotizing fasciitis and streptococcal toxic shock syndrome can be treated with the combination of clindamycin and intravenous immunoglobulins. The drug of choice for group B streptococcal infection is either penicillin G or ampicillin. Some strains may require higher doses of penicillin G or combination of penicillin G and an aminoglycoside to eradicate the organism. The group D streptococci involves enterococcal and non-enterococcal strains. The enterococcal infections caused by group D streptococcus are treated with penicillin or combination of vancomycin and aminoglycoside. But there are some vancomycin-resistant enterococcus VRE, so they are treated with linezolid and daptomycin. And non-enterococcal group D streptococci, for example strep bovis, are not highly resistant and they can be treated with penicillin G. Viridin streptococcus is treated with penicillin. The peptostreptococci can be treated with penicillin G. Prevention. Rheumatic fever can be prevented by prompt treatment of group A streptococcal pharyngitis with penicillin G or oral penicillin V. Use of amoxicillin preoperatively in dental procedures can help prevent risk of infections. The rapid screening test for group B strep antigen is an also important tool for preventing the infections. There are no vaccines available against any of the streptococci except for strep pneumoniae. There were a lot of information that we covered today in this video. But how can we end the video without reviewing this information? So let's review everything really quick. The organism is Streptococcus. It is responsible for causing diseases like pharyngitis, impetigo, cellulitis, sepsis, scarlet fever, streptococcal toxic shock syndrome, rheumatic fever, acute glomerulonephritis, and endocarditis. Transmission occurs by gaining access to blood or tissue, and it also depends on different types of diseases like pharyngitis can transmit via respiratory droplets. Host are the human beings. Primary locations are skin, throat, including oropharynx, gut, and genitals, because they're part of the human normal flora. And they are diagnosed on the basis of gram stain, microscopy, culture, catalase tests. They are treated with certain antibiotics like penicillin, clindamycin, vancomycin, erythromycin, and amoxicillin. And that's it for today's video. I hope you enjoyed it. You've learned something. If you really did, give this video a big thumbs up. Comment down below in the comment section. And also, if you want to connect with me on my socials, I've got my Instagram, I've got my Twitter, and I do upload blogs. See you soon in another lesson video. Assalamualaikum.